Peelaerts. I'm an MSA researcher. I also work with Dr. Patrik Brundin, and I will be the moderator for this Q&A for the research panel. So in case you missed this morning's session, just for housekeeping, again, these are the questions that you can submit at the Q&A button below at the screen. Please only send questions because they will be used for educational purposes. We cannot discuss any cases or treatments, so if you have specific questions, please consult with your healthcare provider. So again, as I mentioned, please send in your questions. We will try to answer most of them. If some of the questions are not addressed in time within the, I think, 45-minute time frame that we have, we will try to address the questions offline afterwards. So again, thank you, Patrik, and all of the other speakers. I think this was a very interesting session. And Patrik, I think you laid it out very clearly, nicely. So thank you for that. And maybe I think it would be interesting to go back to the start. I think you started your presentation by starting or stating the origins or the state of the art with regards to the origins of multiple system atrophy. And we've gotten a few questions about genetics already, and it seems or people are asking if you could again discuss maybe what the difference is between familial and risk genes. So what is the difference between those two? Since you mentioned there are no risk genes, are you assuming that there are? And so is this important for multiple system atrophy?
Right, I'll give that a try. So familial forms of disease are ones where there's an inheritance that you can understand. So for example, the mother or father has the disease, and then there is, for example, a 50% chance that the offspring, son or daughter, gets the disease. Some inherited forms of disease are linked to the sex chromosomes, so they show a slightly different pattern of inheritance. There are also those familial forms of disease that require that both the mother and father have the defective gene, and that you are 1 out of 4 in the offspring that's unlucky that get the 2 bad copies from the parents. But let's put all that to one side, because as it stands today, there is really no clear familial gene for MSA. Then comes the question, what does weak genetic risk mean for MSA? And you heard, I think Tony Lang said it this morning in his presentation, that there is really no strong hint, even when looking for weak genetic risk. And I think I repeated this also. But what does it really mean, weak genetic risk? Well, it means that we all carry lots of variants in our genome. So there are, we call it single nucleotide polymorphisms. So there's small variations in the DNA, but that DNA doesn't all translate into variation in the actual protein. Most of that variation in DNA sits in what has been called the dark matter of the DNA that doesn't actually make proteins. In fact, about 98%, I think, of the DNA that we have doesn't make proteins. And once upon a time, they even called this junk DNA because people thought it doesn't do anything. Now we've learned that this dark matter, the 98%, regulates the production of the proteins that's made then by the remaining 2%. And in diseases, if we now speak in general terms, all diseases have genetic variants that make you more or less susceptible to get the disease. Now, they might have a very, 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 very small effect. And in Parkinson's disease, there have been, the current count stands at around 90 different places, mostly in the dark matter of the DNA, that regulates the risk of getting Parkinson's, but only regulates it a little bit. So let's say lifetime risk is about 1% for Parkinson's. Well, if you have the risk regulator in the dark matter, you might have a 1.2% risk. So it doesn't have a huge meaningful, it doesn't have a huge meaning for the individual, but it has huge meaning for the scientists, because we now can learn from what genes are regulated by those variants in the dark matter. We can then learn what might be going wrong in the disease. So in Parkinson's, for example, we've learned that the immune system seems to play a role, because lots of the dark matter variants that regulate risk are regulating immune system. Now, Why did this long-winded introduction? Well, with MSA, there aren't any clear single nucleotide polymorphisms that clearly, consistently are linked to disease risk. That might be because the studies haven't been large enough. So to really detect such small effects, you need, like some of the Parkinson's studies have, 10,000 people with Parkinson's and 90,000 controls. And then they can see these tiny, tiny effects. So I think we have to be careful and say that currently we don't know clearly a genetic risk landscape for MSA, but it might be because the studies have been too small. And frankly, there's almost nothing in this world except potentially car accidents or trees falling on your head that isn't related to genetic risk. I mean, genetics plays a tiny role in almost everything. And genetics plays a role also in how susceptible you are, or we are, to infections. So we might get to that later. Yeah, so that was a long answer. I don't know if I really answered the question. Was that what you were looking for?
I think you answered the question. So maybe this is an interesting segue into this next question here. So a question from another person in the audience is, why do they think the MSA type in Japan is different? Is this where genetic risk would come in, or is this more related to the environment, for instance?
So I'm going to give a quick short answer, and then I'm going to hand over to the others on the panel and hear what they say. My quick short answer is it could be either or both. I mean, there can be, we know there are amongst ethnic groups slightly different genetic landscapes. So that could contribute to this, but it could also be an environmental factor. It could be a dietary factor, type of infection you get, or type of toxin that's present in the environment. I don't know what Horacio says, or Tony, I think, is still here. And do you want to, I'm looking at the screen, who's actually visible. Edwin is here, so go ahead. What do you think?
Well, I was going to say, as we understand it, the aggregation of alpha-synuclein is highly related to misfolding, and there are multiple ways that alpha-synuclein can misfold. So it may be in the genetic background that influences the way in which your alpha-synuclein chooses to misfold, may make some difference in the variable, what we call phenotype of the disease.
And it could also, that could also not just influence whether or not the misfolding occurs and the cells manage to handle the misfolded protein, but it may also influence where in the body it actually starts.
So yeah, Patrik, maybe you can discuss a bit more about what the role is of different protein aggregates in multiple system atrophy, or PD, maybe even DLB in the different synucleinopathies. How is this important for multiple system atrophy, you think?
So I think this is a question I'm going to let bounce back to Wouter. So Wouter had a seminal study about 5 years ago where he actually studied aggregates from different types of diseases, and he's continuing to pursue this. I'll introduce Wouter's answer by saying that in the different diseases, the aggregates, although they in dementia with Lewy bodies, Parkinson's disease, and MSA are all primarily made up of alpha-synuclein, the shape at the molecular level of the aggregates is different, and their properties, which was hinted in my talk, their properties are also different. And maybe, Wouter, you can explain what you did when you had this paper, which was published in Nature 2015, I think.
So yeah, I mean, of course, I can also say something about this. So we had this idea maybe 7 or 8 years ago that the shape of the aggregate, the shape of synuclein strains, as we call them, might influence the disease phenotype and disease progression. So what we have found is if you take these aggregates out of the brain of a person who has, for instance, Parkinson's disease or multiple system atrophy or dementia with Lewy bodies, and you put them under a microscope, you will see that the shape of these aggregates are actually quite different. And I mean, aggregation and protein misfolding, I think, is an incredibly abstract process. So sometimes I compare them to the way how, for instance, eggs are boiled, because an egg is pure protein. And as with these aggregates, you can actually boil an egg, which will cause the protein inside the egg to aggregate and accumulate and become sticky. And this is what also happens with alpha-synuclein in the brain. So in our brain, we have about 30,000 different proteins. In our body, we have more than 50,000 proteins. And so one of those 30,000 proteins in the brain, alpha-synuclein, for reasons we don't yet understand, actually starts to accumulate and aggregate. And just as with eggs, these proteins can accumulate or aggregate differently. Like you can boil an egg, you can make it sunny side up, you can make a scrambled egg. And for many people, these eggs will taste differently and they will look differently. And this is also the case for synuclein protein. They can aggregate differently. And because they do so, they will also acquire different functionalities. So the way how the protein misfolds and the way how synuclein misfolds and forms these clumps, these sticky proteins, will cause the aggregates to cause different clinical phenotypes. And so when we injected these aggregates, for instance, in the brain of different animal models, we found that these animals started developing either Parkinsonian or MSA-like phenotypes. So this showed, I think at that time, for the first time, that the way how these protein clumps form, that they can determine the disease outcome. And now we are, of course, looking farther than just this, because we've also recently started looking at how these protein clumps might influence multiple system atrophy on its own. Very recently, there was a really interesting paper from a group in England where they looked at MSA aggregates by using a microscope that's called a cryo-EM, so they can look at the atomic level of how these strains or how these aggregates are built. And they found that indeed in brain of MSA patients, they indeed confirmed that in MSA you have a different type of aggregate as for instance the one that is found in Parkinson's disease. But also interestingly, what they found is that you apparently have different forms of these strains within a given disease condition. So for multiple system atrophy, there are also multiple different types of strains. And this is also what we're now seeing in, for instance, animal models of multiple system atrophy. If you challenge them with different types of clumps, these MSA animals will also develop different MSA phenotypes. So there are many different ways in which these aggregates or these clumps can influence disease progression. So this is how Patrik bounced this question back to me. So that's the.
Because it was such a difficult question, I didn't want to even try. I think you're doing great though.
So that might be the role of these clumps in the brain of people with MSA. So maybe we can continue, because I see we also have a lot of questions about UTIs. Also this morning we had many questions about UTIs and how this might be involved in the disease process. So one of the questions here is there any research about the percentage of MSA cases that have UTIs versus other infections? I don't know if there's anyone who would like to address that question.
We could maybe add for the clinicians on the panel, do you have a sense, or would you know of a study about how many patients who already at the time of diagnosis have a history of many UTIs? You know, UTIs is a common phenomenon, but do you have a sense of, is this a very common thing that they can report, 5 years ago I had a lot of UTIs, or is this just anecdotal?
I don't think there's any evidence, to be honest with you. As you say, though, the problem is that urinary tract infections are extremely common in the general population. And so I think it's going to be very difficult to show a significant difference. I think the other problem is that once some of these diseases become more frequent, for example, Parkinson's disease gets a lot of bladder dysfunction and urinary tract infections, and yet those that get a lot of urinary tract infections, to my knowledge, don't develop MSA pathology. So there's something still very unique about the MSA person. If urinary tract infections predispose, they still have to have something much more in addition to just having a urinary tract infection. I don't think that was your implication. I think there's got to be that plus probably a whole bunch of other things. Urinary tract infections don't explain the location of the pathology, why the oligodendrocyte is particularly affected, and how the synuclein gets into the oligo as opposed to the neuron. So I think it's one piece of a very, very large puzzle, probably.
Yeah, I think we wouldn't even propose that every type of urinary tract infection predisposes to MSA. There must be something else or something in the individual that's affected. And there might be this unlucky situation where under certain circumstances during the urinary tract infection, these very bad aggregates form. And there are hints from the literature in animal experiments that perhaps oligodendrocytes are more prone to take up certain type of aggregates than others. I mean, that's a thought or an idea. So as Wouter mentioned, synuclein aggregates can look different, and perhaps it's only certain types then that will be taken up by the oligodendrocytes. And also another idea, yeah, go ahead.
I guess the other issue that needs to be addressed and aired here for the people that are listening is that neurodegenerative diseases are also rather selective in the location of where the pathology is. So there are very important areas that are more predisposed to pathology than others. And so what is it that allows that selective vulnerability, and how does that fit with the hypothesis as well? So we have to explain that side of things too.
And another thing I also think we should bring into the discussion is we call it MSA, it's also subclass MSAC, MSAP, but there's a wide variety of subgroups or like a constant range between these types of pathologies. So the individuals who have MSAC may actually have a totally different starting point of the disease. We don't understand it, but to those who have MSAP.
You know, something to comment is that perhaps the difficult thing is the chicken or the egg. I mean, we do know for certain that the involvement of the bladder is significantly more pronounced in MSA than in Parkinson's, right? Like what we said before, if a patient with early motor signs or very mild motor signs has urinary retention, has bladder retention, that is almost diagnostic of MSA, because unless there is a prostate problem, patients with Parkinson's early on almost never have urinary retention. So one could speculate, following Patrik's suggestion, that it is the early involvement of the bladder through the ONUF nucleus that could be one of the first targets in the MSA process that impairs the emptying of the bladder, and the emptying of the bladder being impaired then makes you susceptible to more infections. So it may be that the process starts as a regular synuclein-related neurodegeneration of the spinal cord, of neurons in the spinal cord, and that gives then the infections that could, like any alteration of the microbiome in the bladder or in the gut, could then enhance that process. I would emphasize one more thing. What I find fascinating of this is that, look, not many years ago, the idea that Parkinson's disease, and I'm not talking MSA, the idea that Parkinson's disease began outside the brain was heresy. I was throwing tomatoes in the things. I mean, people were laughing, saying, why would you say that? I mean, why would it start outside? This is a late complication, not an early complication. When I think that paradigm has changed completely. Surprisingly, though, that paradigm has taken hold for Parkinson's. But in MSA, because of the idea that the postganglionic is not as much affected, that is not that clear. And all the studies from the vagus transmission of synuclein from the gut, all those are from Parkinson's. However, there's no reason to believe that the same thing may not occur in MSA. It's just that maybe the original misfolding, like Wouter described, could be a fusilli instead of a spaghetti. I think that you had used pasta metaphors, Wouter, when you rather than the egg, right? So one could perfectly think that, you know, my feeling, and of course it's way to be proven, is that this is not that much different to other proteinopathies or even tuberculosis. You know, tuberculosis you can have tuberculosis in the brain, tuberculosis in the lung, tuberculosis in the kidney, or in the bone. For 100 years, these had different names, different diseases. Now we know that it's all tuberculosis. It's exactly the same. It just matters how the host reacts to that accumulation. So the example, maybe because I'm a very simple guy, but I think that the example could perfectly apply to MSA. And I would even say that it's not just the bladder. I think it could start in the bladder, in the gut, in the heart, in any peripheral organ, and for all we know, may even start in the skin. The idea that any neuronal structure that if there is inflammation around or something happens with the rest of the inflammatory process, starts the accumulation there, and then there is neural transmission that goes all the way to the brain. We studied it in the vagus. And of course, the bladder doesn't have the vagus, but it has other nerves. So this synuclein can be transported through these cables. Sympathetic nerves may do it, sensory nerves may do it, any afferent nerve may do it. There's no reason to believe that they cannot be highways to the brain. So starting, in fact, the next frontier, and I had no idea about the infection, but we connected with a number of urology centers, and we told them, look, if you have patients with urinary retention that do not have a spinal cord lesion and do not have MS, we want to see those patients. If they don't have a big prostate, they don't have, those guys may have very, very early on synucleinopathy. So I think that that's the next frontier. We did the same with the heart and the same with the gut. What does the bladder have that others don't? My feeling would be that it's the same.
I totally agree with you. Actually, there was an interesting question from somebody. I don't know, can you see the questions in the chat, Wouter? Have you got the chat window open also? Or are you looking at the ones that came in earlier today?
I'm not sure which question you're...
So at the bottom, no, there was one question about myeloperoxidase, and you know, this myeloperoxidase inhibitor that's being tested now, and we heard earlier today that this clinical trial, yeah, you mentioned it, Horacio, in your talk. So I just want to add. I think the question is where does it
got into it, but I still don't know how that happened.
So the question is here: where does this fit in with alpha-synuclein pathology and the role of myeloperoxidase? So the myeloperoxidase is highly enriched in the white blood cells, the neutrophil leukocytes that we talked about. And just want to say a couple of words about that, because now we're talking about MSA, but there was a paper that came out just a month ago looking at polygenic risk score in Parkinson's. And the method basically was a lot of bioinformatics. So they look at many, many genes, and I think 450,000 people or something like this. And they look at then the combined risk of getting Parkinson's, but they then look also at what genes might be interacting with each other. And through their bioinformatics, they can identify gene families that work in certain functions and then seem to contribute to risk for Parkinson's. And the number one family that came up in this study, which comes from a large group of authors, Andrew Singleton is the senior author, the number one family was related to neutrophil degranulation. That's actually the process of when these white blood cells release their granulars as part of the immune defense mechanism. It's also when they release those nets that I mentioned, that's when their granular are being released. So this is, you know, done totally unbiased on huge numbers of people and huge numbers of genes. And for some reason, genetic variability in that pathway is important in governing Parkinson's risk. So myeloperoxidase is highly expressed in white blood cells. I'm not saying that it is myeloperoxidase only in white blood cells that is relevant in these therapeutic trials, but it is a rather stunning coincidence that we're now in a clinical trial for MSA with a drug that affects or inhibits myeloperoxidase. And it'll be very, very interesting to hear the results. I think the results will come out about a year and 2, 3, 4 months from now, because
the trial was fully recruited, which I think you mentioned, Horacio, this summer. And it's a 48-week trial. So there we'll test the hypothesis whether inhibiting the enzyme that's involved in this immune system function in any way can slow disease progression. The problem with these sorts of trials, though, is, of course, they are taking individuals who already had the disease for some time. And these events may be very, very early in the disease process. And it is possible that one has to identify people who, like Horacio described, come in with just one symptom and it's not even MSA yet, and then try these treatments there. But that's something that will be done many years from now. I don't know what you are all thinking about the myeloperoxidase inhibitor trial.
I think I'm looking forward to results as expectantly as you are. I think it's a very interesting possibility. I would emphasize to the listener, though, and I think you probably implied it as well, but maybe we need to state it, that disease-modifying trials are looking for something different than symptomatic therapy. Symptomatic therapy, by that we mean symptoms improving. So symptoms of slowness, for example, improving to levodopa, and we've said that can occur with multiple system atrophy, but to a lesser extent than in Parkinson's. Generally, we think that disease-modifying trials will not improve symptoms. It doesn't mean they can't, but most of the time we think that the effect, if successful, will be a slowing of the progression of the symptoms. And so that's very different. So patients coming into these trials should not expect that their symptoms are going to get better. At the best, they may stabilize. More likely, they will slow in comparison to patients not receiving that treatment. And that's why we have to do these trials comparing an active treatment to a placebo treatment in a double-blind fashion.
Sure, that's what I tried to, Tony, thank you for re-emphasizing that. That's what I tried to explain in the talk about clinical trials, the difference between symptomatic and disease-modifying. In fact, like Dr. Lang is saying now, to use a metaphor, if you use anti-cancer treatment, you can even feel worse initially. So the idea of disease-modifying is not for symptoms, it's not to immediately make you feel better, but to try to address the root of the problem and slow the disease, whereas symptomatic treatments should make you feel better quickly. And if they don't make you feel better, there's no reason to continue them. Whereas in the disease modifying, you could have some side effects that you may need to tolerate.
Okay, thank you. So I see we only have 15 minutes left, I think. So maybe we can take a few more questions from the chat. So one interesting question here as well is, are there any genetic or clinical characteristics that differentiate slow versus progressing MSA patients? Maybe Dr. Kaufmann, if you could comment.
Short answer is no, I guess.
Yeah, please, no, I will, you know, we always learn not to talk about zebras. You know, that's a line that medical students and older doctors we use when, you know, there are always rare cases or cases that do not follow the actual pattern. And of course, they, by the fact that they are so different, that's why they are so interesting. You know, like zebras are interesting. And, you know, I'm still digressing. You know, one thing is statistics and the other sometimes is biology. You see, if you cut the head of 100 people and one survives without the head, you're going to say that that's not statistically significant. Now, I'm very curious to see how that guy without the head continues living. I don't care, he's a zebra, right? So with that said, I will throw the following to the panel. We had a patient, a very nice man, started young, that I followed for many years with young children, and we almost became close friends. And he was an advocate and a speaker for MSA. He died after like 14 years, or if I remember, or more of the disease. And his neuropathology was typical MSA with glial cytoplasmic inclusions, textbook MSA. Now, he turned out to be positive for the LARC mutation. So I just sent that because it's a zebra. Nobody is a movement disorder, but of course, nobody paid attention to that report. I just sent it to Patrik and I think to Tony as well. Is this a zebra? Most likely it's a zebra. Or is it telling us that, the big studies of LARC with thousands of patients, like the people that cut their head, say that there's no connection. But here's a typical case, nothing atypical of it, with even neuropathology is typical, and he has a mutation that is supposed to be associated only with Parkinson's disease. So what would be your first, what does this mean? I have no idea.
Well, I'd comment that LARC2 mutations have in fact been associated with other pathologies. They've been associated with PSP and tau pathology. They've been associated with a bland neurodegeneration without protein deposition, maybe some TDP43, but not much, I don't think. And there is one case of corticobasal degeneration associated with it as well. Now, my take on that corticobasal might apply to yours, and that is we know that a lot of people with LARC2 common mutations never manifest. So they are asymptomatic carriers of a mutation, and they can live to a ripe old age and have no pathology at all. Only about 30% of patients with the common mutation actually do manifest. So you could argue it that way, totally unrelated coincidence, but you could also argue that there are these cases where we think the pathology is different and it is the gene contributing or causing. I'd like Patrik's response. I don't know.
So first of all, we should explain to the viewers. So LARC2 is a protein that is mutated and associated with Parkinson's disease. So it's actually the most common monogenetic form of Parkinson's in the world. And in some populations, very common in North Africa and Israel, amongst Ashkenazi Jewish people too. But as Tony said, it only penetrates, although it's a dominant gene, only penetrates in 30 to 80%, depending on age and which population it is. So it means that some people with this mutation will live to a ripe old age and die and never have Parkinson's. But also the variety of pathologies that you can see in people who develop Parkinson's disease and have LARC2 mutations is quite wide. Not all of them have synuclein aggregates. So it is a very strange thing. And that's as a final point with LARC2, that gene is also associated with Crohn's disease, an inflammatory bowel disease, which of course makes you think about the gut and inflammation and Parkinson's. So remember, risk genes, for the most part, only tell us how likely it is you're going to get the disease or not. They don't tell us that much about how quickly the disease will progress or how poorly you will do, with the exception of GBA, I guess, or another.
So your initial interpretation, Patrik and Tony, which I also thought, is that there's a possibility that the LARC mutation was never expressed, and he had bad luck, had a LARC mutation that didn't express and developed MSA. That's one parsimonious explanation, right? The other
that's a possible explanation. And you say I had bad luck, but it may also have had a lifestyle or a diet that did include whatever the trigger is that is required for 40 to 80% to develop Parkinson. I mean, clearly there are people who don't develop Parkinson's. And I don't think we're saying anything out of line by saying that one of the two founders of Google carries a mutation. So Sergey Brin has been very public and open about this. He has a G2019S LARC2 mutation. His mother has Parkinson's, and he's heavily invested in Parkinson's research for this. But Sergey Brin does not have Parkinson's disease. He knows that the likelihood is 40 to 80%, but there might be things in his lifestyle and the lifestyle of everybody else with this mutation that can affect risk. And maybe this individual then developed MSA for a different reason that had nothing to do with that pathway.
Sure. You could turn it on its head just to show people how complex biology is, and even point out he lived longer than many, many patients with MSA. So maybe his LARC2 mutation somehow protected him in some way too. You have no idea. That's the problem.
Sure, absolutely. There's a question, Wouter, about animal models from somebody called Sonia. What's your favorite MSA mouse model and why? And any work on organoid models? Maybe I can just give a quick answer there that I don't think any of us should have a single favorite model. Each animal model can help us answer specific questions and they complement each other. But I'll just quickly give an introduction to the types of animal models that exist. There are those animal models that use toxins to destroy parts of the brain that die in MSA, but they're the kind of more older type of models that we used before because they don't really mimic the disease process. Then there are many animal models that are based on alpha-synuclein. One way of creating an alpha-synuclein animal model is to make a genetically modified mouse. We call it a transgenic mouse, where synuclein is expressed in high levels, particularly in these oligodendrocytes, the ones that form the aggregates. Another way is to inject a virus into the brain of a mouse, where the virus targets the oligodendrocytes, and once inside there, it makes alpha-synuclein and you can get aggregates. And a third way is actually to do what Wouter talked about before, which is to take the misfolded alpha-synuclein protein, be it a scrambled egg or a fried egg, or whatever it is, you take that protein and inject it into the brain, but you take it from a patient who had MSA. You generate that, or you capture it from such tissue. And then you get yet another type of animal model. And the question whether there are organoids, I'm sure there are. There's certainly work on organoid models. That's when you grow brain cells in a dish. And because it says organoid, it's organ-like models. So it's not a real brain you're growing in a dish. But the aim today, and what people are beginning to achieve, is to create little clusters of cells that contain the nerve cells, the supporting glial cells, the astrocytes, the oligodendrocytes that are affected in MSA, and people are also trying to introduce these immune cells, microglia, into these organoids. I think these are great models because you can use human tissue. You can start with human stem cells and make these, but they have the shortcoming. They are just a clump of cells in a dish. They don't have contact with the rest of the body, with all the endocrine regulating factors, the immense immune system that the body has. You can't recreate that in a tiny dish today. So, Wouter, what's your favorite model?
Well, that's very hard to say because, I mean, I can also add that animal models for multiple system atrophy are far not as developed as the animal models for Parkinson's disease. And I think we still have some work on that in that regard, creating better animal models to understand multiple system atrophy better. So I would say my favorite animal model would mostly depend on how good we understand current models and how well they are available so that other research labs can use the same type of model and can validate the same type of research. So I think it is something that the research field is lacking. I think we should have more MSA models, as in the case of Parkinson's disease, and as many of you have mentioned already, there are many different similarities, but there are still many things we don't understand. I mean, also in an animal model, it is very difficult to express and propagate this synuclein pathology in oligodendrocytes. Like if you would take the aggregated material from an MSA brain and inject it in a mouse, you actually get aggregates in neurons. And so this is also one of the things we still do not understand. And it's also something that Dr. Lang also mentioned earlier in this chat. There's probably a multiplicity of factors and risk genes and maybe even environmental triggers that we currently still don't understand. And if we could apply these to our animal models, this would be wonderful. This way we could create an animal model that would have most, not all of the features of multiple system atrophy that would also help us in bringing new drugs to the clinic. But as of today, I think it's mostly something that I think the research field has to I think we have to create more and better MSA models. That's for sure.
And we might be modeling different phases of the disease. So the very beginnings of the disease process might need a certain type of model. Manifest disease might need another type of model. And I think that's where we are failing, actually, scientists, that we view a model as a model of the whole disease process, which it isn't necessarily. I just wanted to bring up a question I saw about from Blake. Blake says, can the virus of Rocky Mountain spotted fever cause the misfolding of the cells? Or I think it's the misfolding of the protein in the cells. I personally don't know the Rocky Mountain spotted fever very well, but I do know there are other viruses like West Nile virus that have been shown to upregulate synuclein in the brains of animals. So West Nile virus is typically mosquito-borne virus. And if you inject it into a mouse, in some mice, it'll go into the brain and cause a horrible encephalitis, and the mice will die. Now, if you inject it into mice that don't have alpha-synuclein, because you can genetically engineer mice so they don't even have this protein, then it will always go into the brain and cause encephalitis. So a scientist in Colorado, a virologist called David Beckham, he's not the soccer player, but he has the same name. David Beckham in Colorado has shown that, his thinking is that the alpha-synuclein has a role in the brain in limiting the entry of viruses, such as West Nile virus. So that's why in the absence of synuclein, the virus gets in and kills all the mice. And in the presence of normal levels of synuclein, only some mice will get it into the brain. So that is interesting, right? So it makes you think, oh, maybe this is the daytime job of alpha-synuclein, because obviously the protein doesn't exist just to
welcome to the
MSA.
channel. This presentation is called MSA, Speech, Language, and...
So that, I think that's our...
Is that a cue?
So
I'll just finish off by saying there was a paper this week in Lancet Neurology, patient in Israel who was infected with COVID-19 or developed COVID-19 and developed an acute Parkinson-like condition. So it's actually published in the October 1st issue of Lancet Neurology, but it's online. And the patient was negative for LOCK2 and GBA mutations. They have a PET scan showing reductions of dopamine markers in the putamen. And that's kind of weird, interesting. That's not Parkinson's, but it's certainly a basal ganglia condition. And there are two other such reports, case studies. They're zebras, right? The zebras, but they're interesting.
maybe I would ask you, the three of you, Wouter and Tony and Patrik, to also comment that the other maybe elephant in the room that we are not talking about, and is a concept that Patrik also pioneered, and is the idea of the synuclein misfolded species being a prion or prion, right? Which of course generated not only a lot of interest, but also some fear, you know, the difference between an infectious agent versus a contagious agent. And as you're well aware, it appears, I mean, data shows that it's only the synuclein, the MSA synuclein species that actually cannot be sonicated or right? I mean, I think you would, I'm interested in your opinion on that. I mean, Patrik wrote extensively about it, and of course, it's called prion-like, something that the discoverer of the prions hates, right? Because he says, you don't call this virus-like. It's a virus, it's a virus, it's a virus. So if this is a prion, is a prion, is a prion. It's not prion-like. So I would like you to comment on that, because people ask those things, and what is your take?
Well, I can maybe add here. So, right, if you would ask Stanley Prisoner, so the man who actually won the Nobel Prize for discovering the prion, so he would say that synuclein is a prion. But I think here the discussion is probably mostly about the nomenclature and about how we define synuclein and how it functions. it has many of the features that a prion has, synuclein. But this is mostly from animal models, and this is mostly from what we see in a scientific experimental setting. What we also know is that in a more clinical setting, that synuclein, I mean, it's very hard to show that synuclein or aggregates of synuclein can spread between individuals. This is something that has not been shown. And so in that regard, I don't think we can say that it would spread from one person to another person. And that's a really important difference. Other than that, it can misfold within a person's body. It can aggregate, and those clumps can cause the misfolding of a second protein that will then cause a quote unquote infectious mechanism and a propagation or a spreading of the disease that resembles that of a virus. But of course it is not a virus. So it has many of those features, but I think there is no proof that there is individual spreading of synuclein aggregates. But from a scientific perspective, I like to approach the functions of aggregated synuclein the way that a prion has been described.
And remember, in all our experiments, we utilize very high concentrations of these aggregates. So if it's in a cell culture or in an animal, when we're injecting, it's typically at high loads. And under those conditions, you always get pathology, and it always spreads along nerve tracts. But in the real world, and tying into what Wouter said about there has never been a demonstration of a case where there's communicable disease, one individual to another. So in the real world, if somebody for some reason ingested some misfolded synuclein, I got into the gut, I think it has to go through many barriers before it would reach the nervous system. And if it did reach nerves in the enteric nerves of the gut, those nerves perhaps can degrade it, because we have autophagy, and this was mentioned earlier today with one of the clinical trials that's a drug that stimulates autophagy. Autophagy can take care of misfolded proteins up to a point. So it's only when it's overloaded and overburdened that it would trigger a spreading disease. And a young, healthy individual, perhaps we're all exposed to these misfolded proteins in our daily lives, but they don't do anything because we degrade them.
Thank you.
Okay, maybe I should suggest an encore.
Yeah, another time.
It says, this is the end, I think it says. In the chat, we need to close. I got a private message from Defeat MSA.
So the speech, language, and swallowing talk, I think, has already started. So I can ask people to go to the next talk, in the next session, and then the questions that haven't been answered here that were posted in the chat box, we will try to address them and post them to you later on. So thank you very much. Thank you to all of the speakers and the panelists to be here. I think this was a very interesting discussion, and I'm excited to hear more about the other talks here today. Thank you very much.
Thank you.
Thank you.